Right, today we are going to talk about the one more monkey, that is the fibroblast. And one of the important function of the fibroblast is to produce collagen. So we'll talk about fibroblast and its capacity and mechanisms, how it produces collagen. Collagen is rather surely, it is the most abundant protein present in our body. Collagen is the most abundant protein present in our body. Your 25% of dry weight is just collagen. In human body, 25% or slightly more than that dry weight is just collagen. So you can understand that if collagen is not there, rather I must say that if collagen was not there, human beings will be just a lump of cell with some neurons interconnected. These are the collagen which provides strength to the bone, they provide strength to the cartilage, and they provide strength to the most of the connective tissue, right, with their fibers. And collagen can be produced by many different types of cells. Mostly it is produced by fibroblast, right, but endothelial cells can also produce collagen, osteoblast can also produce collagen, chondroblast can also produce collagen, right. Why? Because fibroblasts are making the collagen for all the connective tissue. Then to produce collagen for bone formation, osteoblasts are producing collagen. To provide collagen for cartilage formation, chondroblasts are providing you collagen. Is that right? Even collagen which is around the, you can say, collagen fibers which are making a lot of connective tissue near the smooth muscles, even smooth muscles under certain circumstances can synthesize collagen. So collagen can be synthesized by many cells, but here we will take the most typical example that how a fibroblast produces collagen. Let's suppose here the fibroblast. Okay, let's put it on the side. We make a cell here. This is our very synthetic monkey, you can say. This is our synthetic monkey. Yeah, he's very happy. You're going to understand about it. Alright. Now, it's a fibroblast, how it is going to work and how it is going to make collagen. You know, collagen is a protein, and protein synthesis require messenger RNA, and messenger RNA is coming from transcription of DNA. So the real story starts from the real control room in the cell, that is the genetic information, because the secrets of synthesizing collagen are locked into DNA, in the genes which are concerned with the collagen formation. So let's suppose here is the nucleus, right, and here is double-stranded DNA, and here is the gene for, let's suppose these are the nucleotide sequences, and these nucleotide sequences are making a gene for synthesis of collagen. Is that right? How do you define gene? Gene. Who will tell me what is a gene? Because whenever we talk about the protein synthesis, genes story should start from genes, because genes direct the synthesis of peptide chains and proteins, isn't it? And when we talk about protein, the mutant, actually these are the genes which are mutant. Is that right? So what is a gene? Anyone? Genes are sequences of nucleotides within the DNA. Gene is a specific, write it down, it's very important. Gene is a specific nucleotide sequence within the DNA concerned with the formation of specific RNA. Right? The modern concept of gene is a specific sequence of nucleotides in DNA. Gene is a specific sequence of nucleotides in DNA which is concerned with the synthesis of specific RNA. Let's suppose if this is DNA, right? This double-stranded DNA molecule may have many genes. It may have many genes. Maybe the nucleotide sequence here, it can make a different RNA. It may make messenger RNA. Another gene which is present over here, this may make ribosomal RNA. and there's another gene maybe which can transcribe from here, and this may make transfer RNA, and some genes lead to synthesis of small nuclear RNA. So what are genes? You know, within the double-stranded DNA molecule, there are sequences of nucleotides. Within the double-stranded DNA molecule, there are specific sequences of nucleotide which lead to the formation of specific RNA. Is that right? Now it means, now out of these all RNAs, right, this, the only RNA which is translated is messenger RNA. That nucleotide sequence on the messenger RNA is translated into formation of peptide chain. Peptide chain. Are you understanding me, or I should elaborate it further? Okay, I will explain. Let's suppose that I will make it this area a little bit large. This is double-stranded DNA. For example, here is adenine, guanine, cytosine. Then there is, suppose, thymine, adenine, adenine. Is that right? Now, actually, this is a special code. Let us suppose for a special type of amino acid. This is a code. For example, the amino acid which is coded by that is alanine. We are just supposing. And this is a code for another amino acid, and that amino acid is, let's suppose, lysine. Is that right? Now listen. In this way, within the DNA, these triplets of nucleotide, right, are coding for a specific information for a specific amino acid. But actually, DNA cannot be directly translated into amino acid because DNA is present in nucleus and amino acids are present in cytoplasm, and protein synthesis occur in cytoplasm. It means this genetic code which is present over here should be brought out by a special type of coding message, and that message should come into cytoplasm, and in the cytoplasm these amino acids should be arranged to make a peptide chain. Now what happens? That actually from this DNA we make complementary RNA first. Suppose this is DNA. From this DNA we will make complementary RNA. Now you know, when we are making complementary RNA, in front of adenine what comes? Normally complementary nucleotide for adenine is thymine, but in this case there should be uracil. And in front of guanine there should be cytosine, and complementary to cytosine is guanine, and then complementary to thymine is adenine, adenine to uracil, uracil. Now actually this transcribes into this. So special secret code in DNA transcribes into secret code in RNA, and if it is messenger RNA, it will go into cytoplasm. In the cytoplasm, this messenger RNA, I'm just showing 2 points only, but it is very long with lot of codes. Now it will be studied by, what are these? Ribosomes. Ribosomes are special machinery. What ribosomes can do? For example, this is ribosome. Now this RNA code is passing through the ribosome. The function of ribosome is that it is UCG. What is this? UCG. And next code is AUU. Is that right? So the secret in the DNA is transcribed into RNA. RNA comes into cytoplasm, and then this will be translated into peptide sequence. Now UCG is actually for which? Alanine. And AUU is for suppose lysine. We are just supposing. Now what happened? That there are transfer RNAs. There is another type of RNAs. Now these are two RNAs, and this is an RNA. This is called transfer RNA, which is carrying lysine. This is another transfer RNA which is carrying alanine. Now they are supposed to transfer the amino acids at appropriate position in the ribosome and messenger RNA. Now what happens? when ribosome is reading, suppose UCG, then this will fit into this position, right? So it will fit like this, and what will be positioned there? Alanine. Is that right? Then messenger RNA will slip forward. Then this ribosomal complex will be reading this position. And when this will come into this position, then lysine carrying transfer RNA adjust with this code, right? So lysine will be put into next position, and then this is the function of ribosome to put them into bonded position, and peptide chain is formed. This process is what we are doing. The code of, according to the code in the messenger RNA, we are sequencing and making a chain of amino acid. So technically we say messenger RNA is being translated into peptide chain. The codes in messenger RNA, right, are being translated into what? Amino acid sequence in peptide. So in the cell, where are the secrets of protein synthesis locked? They are locked in DNA. Those secrets are, again, this is a DNA. Now what happened? Complementary to DNA, what we make? Complementary to DNA, what we are making? RNA. Then RNA will come into where? Yes, please. Cytoplasm. Then who will start reading the RNA? Ribosome, right? So when ribosome provide a special machinery in which RNA, information can fit, and according to that information, complementary, what is this? Transfer RNA will bring a special type of amino acid into that position. Is that right? So as messenger RNA will keep on slipping through ribosome, with every 3 nucleotide, it will adjust one transfer RNA with a special type of amino acids. And it is a function of ribosome. Ribosome has special enzyme within its structure which can stitch one amino acid with the next one. So ribosome is having the capability to synthesize a peptide chain. But it does not know, ribosome alone does not know that in which sequence amino acids should be arranged or attached. From where the information come to ribosomes? Information come from messenger RNA. It's bringing the message. A messenger RNA is bringing the message of DNA. Am I clear? Now let's make it short. How the protein synthesis start? Every cell and every cell which makes protein by the same mechanism. Either you are making insulin, or you are making growth hormone, or you are making any protein in the body. Mechanism is same. Story will start from nucleus. In the nucleus there should be chromosomes. In the chromosomes there should be DNA. In the DNA there should be genes. Genes should be consisting of nucleotide sequence. Different nucleotide sequence are coding for different amino acid sequences within the DNA. So nucleotide sequences in a gene, right, first they are transcribed into RNA. If they are transcribed into messenger RNA will bring the message of this particular gene into cytoplasm. Messenger RNA will adjust with the ribosomes, and ribosomal machinery will start working on the messenger RNA and keep reading the coding system on the messenger RNA, and according to that allowing the transfer RNA to bring and align amino acids, so that different amino acids keep on stitching there and peptide chain is formed. For example, this is a ribosome. Let me tell you, suppose this is messenger RNA, this is ribosome number, and this ribosome is reading messenger RNA. It has read this point and this point, so it will have maybe 2 amino acids attached. One amino acid is this one, other amino acid is this one. Then for example, these 3 points are coding for star amino acid. Then these 3 points are coding for, let's suppose, another amino acid. These points are coding for still another amino acid. So what is happening? That as ribosome keep on moving, but truly speaking, it's not the ribosomes which move on the messenger RNA. It is the messenger RNA which moves through the ribosome. And ribosome keep on translating the message in the messenger RNA into peptide chain sequence. Am I clear? Yes, please.
The role of tRNA is
Role of tRNA is only to transfer the amino acid, right, for peptide synthesis on the ribosome and messenger RNA. Now, if you really want to learn it so well, let me tell you. We started from DNA, we were having this sequence. Let's suppose we make it simple. This is guanine, guanine, guanine. I'm making it very simple coding, right? And here we have cytosine, cytosine, cytosine. Is that right? When this sequence will be into complementary, what is this? Transfer RNA. So this transfer RNA will have which sequence? Triple G will be transcribed into triple C, and triple C in the DNA will be transcribed into RNA triple G. Is that right? Now it comes, I cannot make it more easy. Here is a ribosome. Ribosome. It is going to read it, right? It's a translator, right? It translates the genetic code in RNA, right, into peptide chain. So when it reads this, for example, this come into, there's a pocket here. When this 3 nucleotide sequence in messenger RNA fit into that pocket, what is this? C, C, C. Is that right? Then it will allow an RNA to adjust with it. Only that RNA will come here which has G, G, G. And that RNA should be carrying, if triple G was for, let's suppose, methionine, then it should be carrying methionine. Okay. Let's suppose if this triple G was for star amino acid, we make it simple, star amino acid, then actually triple G is the sequence in DNA transcribed into triple C in RNA, which will adjust with complementary transfer RNA. The messenger RNA sequence will adjust with the transfer RNA sequence, and this is triple G, and that is carrying star. So you understand how the message from here, message in DNA, is converted into message in RNA, and message in RNA is read by the ribosome, and complementary transfer RNA is adjusted, right? And that brings the appropriate amino acid. Now listen carefully, attention. Another example, let's suppose triple C. This triple C is for moon amino acid. Let's suppose moon amino acid. Now how the moon amino acid, it will code for how the machinery will work. Triple C in DNA will transcribe into triple G in RNA, and triple G in RNA will adjust with what? Triple C in DNA transcribe into, this should code for moon. So wherever in the DNA triple C come, moon amino acid should come in the peptide chain. Is that right? In that sequence. But how the whole system work? Triple C in DNA, what it is? Transcribe into triple G in messenger RNA. Messenger RNA adjust on the ribosome. This will slip into this position. Then triple G sequence will accommodate with transfer RNA. This was messenger RNA, and what should be accommodated here? tRNA. tRNA should have C, C, C. This is triple C and here's triple C, right? And it must carry which amino acid? Moon amino acid. You understand how that triple nucleotide code system in DNA is, right, making complementary messenger RNA will adjust with complementary transfer RNA, and transfer RNA transfer the amino acid in the ribosome at appropriate position, so that then ribosome will make a bond between the adjacent amino acid. So what is the function of ribosome? Number one, ribosome provide a place where messenger RNA can bind. Number two, they also provide a pocket where transfer RNA can bind. And after that, they link the amino acid with each other. In this way, they help in formation of peptide chain. You are very clever. I was supposed to teach something else. but you are studying something else from me. But without it, we cannot understand the synthesis of collagen. Now, for hypothetical purpose, yes, please, you have a question.
One last question. The tRNA, it would be coming from...
Gene. Look, there are some genes which make messenger RNA. There are some genes which make ribosomal RNA, because in the ribosome there is some RNA. Within the ribosome there is some RNA. Ribosome is made of RNA and proteins. There is tRNA also coming from the genes, and small nuclear RNA also coming from gene. Small nuclear RNA remain in the nucleus and help in maturation of messenger RNA. Once messenger RNA is made, later on you will study the small nuclear RNA help some modification in messenger RNA, so that messenger RNA become mature. Mature messenger RNA come out in the cytosol, and when mature messenger RNA will come out, already some genes have made the synthesis of ribosomal RNA and transfer RNA. And transfer RNA which are already present in cytosol, every transfer RNA is already loaded with certain amino acid. So transfer RNA are just amino acid carriers, and they have a prongs like this. On one side they can read the messenger RNA sequence, on the other side they are carrying a amino acid. So if there are 20 amino acids, so there must be at least 20 transfer RNA which will carry amino acids. You are understanding? You will study all this coding system in detail in molecular biology. But anyway, here what I am saying, so ribosomal RNA are made by gene, transfer RNA are made by gene, snRNA are made by gene, and messenger RNA is also of course made by genes. So what happens? That truly the nucleotide sequence on which RNA is eventually translated into peptide chain formation, that is only messenger RNA. Ribosomal RNA is not translated, transfer RNA is not translated, snRNA is not translated. Is that right? It means the nucleotide sequence which are present on these, according to those nucleotide sequence, we don't make proteins. Is that right? But still, all of these also help the messenger RNA to synthesize peptide chain. How? Their small nuclear RNA help the messenger RNA to be mature, which we'll discuss later. Ribosomal RNA help the messenger RNA to stabilize on the ribosome. Transfer RNA help the amino acid to be transferred at appropriate positions of the messenger RNA on the ribosomes. Is that right? So all of them are concerned with protein synthesis. Am I clear? No problem. But which one is truly translated into peptide sequence or amino acid sequences? Messenger RNA. Is that right? So if I say that collagen is made of peptide chains and peptide chains are made of amino acid sequence, it means to make amino acid sequences, we must have messenger RNA. And if we have messenger RNA, then from where the messenger RNA should come? From DNA. So story of collagen formation should start from DNA. Is that right? Am I clear? So what really happens, the fibroblast, the very special monkey, it has to produce a lot of collagen. It means fibroblast in its nucleus activate the genes for collagen. Genes which are concerned with the formation of collagen. Later on I will tell you that collagen is basically made of, collagen molecule is made of 3 peptide chains making a triple helix. This is one peptide chain, this may be another peptide chain, and this may be still another peptide chains. So collagen molecule is basically triple helix of peptide chains. These peptide chains are called alpha chains. What are they called? Alpha chain. Let's suppose this is alpha 1 chain. This is, let us suppose this is alpha 2 chain. Let us suppose this is called alpha 3 chains. So what is collagen molecule? Collagen molecule is basically a triple helix made of 3 peptide chains. Those peptide chains are called which chains? Alpha chains. It means that the genes which should be activated in the DNA of fibroblast should be genes for alpha peptides. Because in the nucleus of fibroblast, there are all the chromosomes and all the genes. Every cell has more than 30,000 genes. Okay, this concept is also important. Yes, I'm going to repeat. I know this is worth repeating. I'm going to repeat. Look, you know, beta cell of pancreas produce insulin, you know that? And you see hair follicle, they produce keratin. Don't look in front area. Right, they are here. So now, this is, suppose this is the beta cell of pancreas. It is producing insulin. And here is a hair follicular cell and it is producing keratin. It means that in beta cell of pancreas, gene for insulin is activated and hair follicle cell is activating the genes for keratin. But the truth is that every cell has every gene. The truth is every cell has every gene. Why cells are different from each other? Because they activate different sets of genes. Now listen carefully. Actually, insulin producing cell has the gene of keratin but does not activate it. And keratin producing cells do have the genes for insulin but they don't activate them. It's a very beautiful concept about the life, that every cell has a full code of life. That is why just simple zygote. You know, you are made from a zygote, isn't it? There was a zygote and then billion, it multiplied and its DNA again and again and it became a multi-billion cell human being. And every time when zygote divided into 2 cell stage, 4 cell stage, 8 cell stage, what is happening? Every time it replicates its DNA very faithfully. Actually, the set of genes which are present in zygotes, they are present in every cell which undergoes mitotic process. So due to this so beautiful replication of DNA, every cell has every gene. Whole genome. Every cell. It's a so immensely important concept and so motivating. You can say it makes me so happy when I imagine this. Every cell has every gene. But the question is that why cells are different? Let me tell you a simple analogy. Let us suppose there is a world-class book in which there are cooking recipes. And you have, let's suppose, 20 girls. And in every book has 30,000 recipes to cook different 30,000 type of meals. And for example, there are 30 girls. Everyone is given this full set of books. Those recipes, 30,000 recipes, are in how many volumes of the books? Yeah, 23 volumes. Let's suppose you call this girl here, you come here. We imagine you are a cell. Come here. Let's suppose this is a cell. Is that right? Now, from mother side, listen. From mother, for example, first we imagine girl and then we go back to the cell. She's a girl. Her mother gives her 23 volumes of cooking books. Her father also gives her 23 volumes of cooking books. Cookery books you call them? Okay. Right, cooking books. Right. Now how many volumes she has? 23 from mother and 23 from father. The volume number 1 from mother, volume number 1 from father. They're slightly different. But for example, volume number 1 is only about the sweet dish from mother and all sweet dish recipes from father. Then volume number 2 has Chinese food from mother side and Chinese food volume coming from father side. Then Mexican food from mother side, volume number 3, Mexican food recipes from the father side. So she has how many, you can say, volumes? 23 from mother side, 23 from father side. Is that right? And one set has 30,000, 123 volume books have 30,000 recipes. And it means 30,000 recipes are coming from mother side and 30,000 recipes are coming from father side. Is that right? Yeah. Every recipe means one gene. One recipe means one gene. One volume means one chromosome. Mother gives how many chromosomes to one cell? 23. Father gives how many chromosomes? 23. Every chromosome has many recipes to cook proteins, to make proteins. Every chromosome has DNA molecule which has many codings how to make proteins. Is that right? So we can say that every cell has 46 chromosomes, 23 from mother side, 23 from father side, and 23 chromosomes from mother side are bringing how many recipes? 30,000. So 30,000 gene from maternal side packed into 23 chromosomes of maternal origin. 30,000 gene coming from the father side packed into 23 chromosomes coming from father side. Am I clear? Right? Now what happens? If there are 10 girls and you give them these recipe books, one of them may be making Chinese food, other may be making some Mexican food, another making the burgers. So all of them will be opening the same, working on the same recipes or different recipes? Different. So all of them will have same material or different material to offer you? No, they have the same recipe books, but they will cook different dishes for you. Cells are like that. All cells have the same recipe plans, but they activate different plans. Is that right? For example, one cell is activating gene number 23 on chromosome number 1. Other cell is activating gene number 200 on chromosome number 2. So both cells will produce same proteins or different proteins? Different proteins, right? Now you have a seat.
What makes them know?
Listen, just step by step. Let's go back. It's worth understanding. Every cell has every gene. 23 set of genes coming from mother, 23 sets of genes coming from father. Is that right? Chromosome number 1, one copy from mother, one copy from father. These chromosomes are homologous to each other. What is their relationship to each other? They're homologous to each other. And on homologous chromosomes, genes which are point-to-point matching, they are called alleles of each other. For example, chromosome number 1, volume number 1, at page number 200 has how to make a special sweet dish. Similar messages on the chromosome number father at point number 200. So those two recipes, one maternal recipe, one paternal recipe, are allele to each other. Is that right? Am I clear? Now, question is that why different cells are different if all the cells in the body have same genetic material? Answer is that all the girls may have the same cooking book volumes, but still in their kitchens they may be cooking different things. They are activating different type of pages of the recipes sets. In the same way, different cells, even though all the cells have the same sets of genes, but different cells activate different cells of different types of genes. So different cells make different types of proteins because different cells make different structural proteins. So different cells are structurally different, and because different cells make different functional proteins, so they are also functionally different. Is that right? So what is the difference in keratin producing cell and insulin producing cell? The difference is not in the genome set. Chromosome number is equal in both cells. The number of genes are equal in both cells. But insulin producing cell activate the genes for insulin. And keratin producing cell activate the genes for keratin. And please don't forget your special monkey. What was that? Fibroblast. It has also the same genetic material. The genetic material in insulin producing cell, genetic material in keratin producing cell, and genetic material in fibroblast, they are all same. Fibroblast will not activate the gene for insulin. It have the gene, but it will not open. It will open the gene to active which have the message how to cook or how to synthesize the alpha chains of collagen. Right? Am I clear to everyone? Let's come back. So what I'm saying, that collagen is basically a triple helix made of alpha chains, alpha peptides, right? So it means to synthesize one type of collagen, cell has to activate different collagen or alpha genes. There are multiple alpha genes. You know how many alpha genes are present? They say there are so many alpha genes for the collagen present in different chromosomes. Is that right? and different cells activate different alpha genes, and they make different types of alpha peptide. So it means that different types of collagen molecules can be produced. For example, one collagen molecule may be like this. This is also triple helix, right? There is another collagen molecule which is like this. That is also triple helix. Is that right? Now these are 3 different collagen molecules, but all of them are collagen molecule. This is also triple helix. This is triple helix. This is triple helix. But it is using the same type of 3 alpha. It is using 3 different types of 3 alpha. It is using 2 same and 1 different type of alpha. So it means there are multiple genes to synthesize alpha chains. Different genes are slightly different arrangement for the amino acid. So how one alpha chain is different from, for example, how this alpha chain is different from this one? Probably a little amino acid difference, amino acid sequence difference. Of course, that difference is because that difference was embedded in the DNA coding. Am I clear? No problem up to this? Let's have a break.